Welcome back, everyone, to the State of Play for 2021. For this year, for 2021, we're going to run a slightly different to last year. We're going to run a monthly seminar series rather than a weekly one. And we'll run the seminar series through till October. And these are going to feature, as last year, the best and brightest researchers telling you what they've been up to and how their research is increasing our knowledge of MND and how perhaps lead us to better treatments. So also this year, now that the COVID threat has somewhat diminished, we're also planning on running a face-to-face research meeting later in the year. So please keep an eye on our social media and websites for details about this. This will be happening hopefully in November. Right, just before we get going, first an acknowledgement of country. In the spirit of reconciliation, MND Australia acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respects to the elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. And I'd like to acknowledge the Wiradjuri people on whose land I am and pay my respects to elders past and present. So today we have two presenters talking about their research and how it's helping determine some of the underlying mechanisms driving MND and how we can actually target these mechanisms to develop new treatments. So first up, we have Dr. Luke Mccallery from the University of Wollongong tell us about how his work on protein accumulation in neurons. Then following that, we have Dr. Alan Yu from the Walter Eliza Hall Institute in Melbourne, who's going to tell us about his recent work in neuroinflammation. So each presentation should go for about 20 minutes, and then we'll have a combined question session at the end. If you want to ask questions, please use the Q&A or chat function in Zoom if you're on Zoom, or if you're in the Facebook Live feed, just leave a question in the comments section and we'll collate all those questions and we'll be answering those as we go through them. So without any more delay, please, Luke, take us away.
Thanks, Kevin. So let me share my screen. All right. Okay. So thanks again, Gethin, for that kind introduction. I'm Luke, and I'm from the University of Wollongong, and I work in Justin Yerbury's lab here as a postdoctoral research fellow. And I'm going to describe some of the research that we're doing here at the moment and where we might take that. So let's get started. So before we go anywhere into the ideas of how a disease might occur, we need to understand what we call the central dogma of biology. And this is the idea that underpins all of biology. And it's basically summed down to there is DNA, which contains all of the information necessary to generate an organism. This information can be copied into a smaller molecule called RNA, which would contain a single gene rather than the 20,000 genes that exist in DNA. And then this RNA is translated into a protein, and the proteins being the effector molecule inside a living organism. And we can understand this sort of like DNA being a library. However, if you want to take information from the library, you can't take the whole library. You take a small book that may contain information that you need in it. And then this information allows you to create an outcome, such as maybe some food or something. And this dogma, this underlying idea, can be affected, unfortunately, by mutations in genes. So if you have a mutation, which may be represented by this star here in your DNA, this will invariably be in the RNA and the protein as well. And just like a library in a book, a mutation is often just a typographic error when DNA is being copied. And this can lead to there being, say, a typo in the book that leads to, instead of a nice hot dog or sausage, you get a burnt hot dog, right? And that's not what anyone wants. And this is sort of what we deal with in cancer and neurodegeneration all the time. So our lab here is interested in the ideas of mutations causing disease at the protein level. And to understand how these might cause diseases, how mutations might cause disease, we have to understand what's called protein folding, because a protein doesn't just form in a shape that allows it to do its job. It's a string of molecules that has to undergo several steps to reach what we call a conformation, which is just a shape that lets it do its job. So a protein would first be created, and it's in this unfolded, unstable, string-like shape, right? Many different shapes can be held there. It'll partially fold, right? It'll get a little bit of structure and become partially stable. And then ultimately, it will reach its folded final stable state. And this can take anywhere between a couple of nanoseconds to up to 10 seconds, which is a long time in biology, 10 seconds. So if a protein wants to transition along this pathway to the final state, there are some unfortunate effects that may occur to it. And so one of these effects is the formation of what are called protein aggregates. And these are when many proteins, instead of folding by themselves, might interact with each other and create a clump inside a cell. And the thickness of these arrows denotes how likely that each protein at a different stage of folding is to transition down a pathway to forming a protein aggregate. And what we sort of understand is that it's these highly unstable, unfolded proteins that are most likely to transition to become aggregates. And this is important for us to understand because protein aggregation is associated with human disease. And this is not by any means an exhaustive list of diseases that contain protein aggregation, but it is probably the most well-known one. So we have Alzheimer's disease, which affects a large proportion of the population, Parkinson's disease, Huntington's disease, the prion diseases like Creutzfeldt-Jakob's disease, and then what we're interested in, which is motor neuron disease. And here you can see pictures, this little gray picture here of what protein aggregates look like if you zoom in really far on them, and then pointing at what they look like in a human sample from someone who has suffered from these disorders. And so we think that understanding how proteins become aggregated, misfolded, and the formation of the aggregates themselves is an important thing for us to understand so that we can come up with ways to mitigate this problem, which is associated with human disease. So I'm not going to belabor on motor neuron disease too much because I think most people here understand it. But what I will sort of give background to is the protein superoxide dismutase 1. This was the first MND associated gene found all the way back in 1993. And since then, it's been a hotly researched thing. We know a lot about it, but we don't know everything that we can. So what we do know is that the protein, superoxide dismutase 1 or SOD1, is an antioxidant protein. So it floats around in your cell and it scavenges oxygen and radical oxygen species. The protein contains copper and zinc, so the copper here in blue, and the zinc here in orange. These two molecules are bound within the protein and they help it to do its job as an antioxidant, and they also help it become stable. One of the things we do know that's highly important for us as disease researchers is that there's over 160 MND mutations that can possibly occur anywhere in this protein. So it's really susceptible to misfolding and aggregation as a result of these mutations. And that's where we focused our research, understanding how we can mitigate the misfolding induced by mutation. So how do we understand SOD1 folding? Well, here's SOD1 freshly formed in the cell as a cartoon. and it's missing its copper, it's missing its zinc, and it's missing these two little red bars here being combined into something called a disulfide bond. So the first step in SOD1 folding is the acquisition of a zinc molecule. The next step is the acquisition of a copper molecule. And then the final step is the formation of this red bond. And this basically makes the protein go from this unfolded, unstable state to a very highly stable state at the end. In fact, SOD1, when it's in this most stable state at the end, can survive being boiled, making it one of the most stable proteins in the human body. However, aggregation can still occur, even in a healthy human. And so the thickness of these arrows denotes how much aggregation we might get compared to these arrows, telling us where the SOD protein is going to want to go. Unfortunately, when a mutation happens in here, the result is typically this, that we get less of a transition from this zinc state to a copper state, and less even to this final state here. And rather what we see is a majority of the protein transitioning down these aggregation pathways to forming aggregates. And that this is associated with disease means that we want to mitigate this. And so we've come up with a strategy to help mitigate this in motor neuron disease, based upon findings from us and others of small molecule drugs that can help SOD1 fold. So the first compound is probably one you've all heard of, and it's called copper ATSM. And this is a compound that helps deliver copper to SOD, helping it proceed down this pathway. The second compound is one that our lab helped researchers from the University of Liverpool in England discover, and it was called Epsilon. And it's a drug that helps this red bars, which represented what's called a disulfide bond, form in SOD1 protein, further stabilizing it. And we've been working on an idea in the Mccallery lab that a one-size-fits-all approach is not going to work for any neurodegenerative disease. And we need to start thinking about things in combination and as polytherapies. And this led to us thinking, well, why don't we use both drugs in combination to see if it can further enhance the effects of each, much like people do in cancer research. And so this is what we hope to see. We hope to see less protein going down these aggregation pathways and more protein becoming fully folded. So to first do that, we have to generate what's called a model of SOD1 MND, where we can model the disease in a way that we can treat the disease, but it's easy and risk-free to us. And so how we do that is we take DNA that encodes for the SOD1 protein with a mutation in it. We attach some extra DNA to it, and this lets us create a protein where SOD1 is tagged with a new protein called green fluorescent protein. This lets us see where the SOD1 is inside a cell, because the cells that take up our DNA will glow green because the green protein is there. And so what this might look like under a microscope is if we have healthy wild type normal SOD1, we can see it's quite nicely spread throughout the cell. The cell looks nice and healthy. But if we have an MND mutant SOD1, our cell has these very dense, bright clumps of protein in them. And these are the aggregates. So we're capable of modeling the disease in a very simple, easy to manipulate system. And this helps us do what's called a drug screen. Now, we obviously have 2 compounds we're testing, so we're not really screening through many, many drugs, but it helps us in our future prospects of doing such a drug screen. And so we've established a method here at UOW where we're capable of taking millions of cells that we've put DNA into that are somewhat of a mixed population. Not all cells get sick when they make an MND protein. Not all cells form protein aggregates. So we have to analyze this in a quantitative manner so we can figure out how many cells have formed aggregates, how many cells did get sick, and therefore can we figure out if the drug has decreased the levels of those sick cells. So we take millions of pictures of cells using microscopes here, very advanced microscopes, and we feed them through artificial intelligence. And the artificial intelligence will automatically, since we've trained it to understand what we're trying to do, it'll automatically sort cells into healthy cells and sick cells. And this way we can quantify the effect of our drug. And so here's just a little bit of information that we've recently seen and that we've been working towards. So this is that, like we hoped, copper ATSM and epsilon do seem to synergize in our model. So here in this graph, what you can see on the left-hand side is the percentage of cells that we measured as sick. And then here is the amount of drug in no particular units. And if we treat the cells with just epsilon alone or copper ATSM alone in blue and orange respectively, we can see we do decrease the number of sick cells. But if both are given at the same concentrations together, we can see we greatly enhance the effect to reduce the number of sick cells in the treatment. And we've found this, I don't have the data here, but we found this is because it does promote the folding of the SOD1 protein, which is what we thought as well. So that's really nice to see. And what we're now doing is trying to publish this information, and we're now trying to use this information to move into what we call preclinical models of disease, so that we can hopefully see this one day move into the actual clinic. But for us, as mostly basic researchers, we want to look at other future prospects. And so one of the things we're trying to do now is we're trying to identify compounds that may help with this step, so the acquisition of zinc, and compounds that may even help remove or clear the protein aggregates that we see. And what we're going to do to perform this is an unbiased drug screen where we use this system we've set up, this AI system, to simply measure millions of cells, but thousands and thousands of different drugs to see if one can be found that may link and synergize with these two drugs that we know now work together. And with that, when we're moving forward, I'd like to summarize what I've talked about today. So we all know motor neuron disease is associated with protein aggregation, and that the potential to remove the protein aggregation or prevent it from occurring may help treat or alleviate disease. We've got two drugs that seem to work together specifically for SOD1 disease. We want to definitely test these as well in models such as TDP43 and FUS. And we are going to move towards looking at thousands and thousands of drugs and drug screens in the future. And with that, I want to put names, faces to the research here, and this is the current and a few past members of the lab here, Justin Yerbury's lab. So there's Justin. I'd like to acknowledge Justin for looking after us all and supporting all the research we want to do. In particular, Natalie and Madison for doing a lot of the copper ATSM work, Victoria for leading a lot of the epsilon work, and Jeremy and Mccallery, who are going to be working pretty much now are working together on moving this drug combination into animals. And thank you for listening to me. So I have to introduce Alan, who's up next, who's going to talk about some pretty exciting research with inflammation. So thank you.
Yep. Thanks, Luke. That's very nice talk. I really enjoyed that. Thanks for the invitation to be here today. Really excited to talk about our research, how we targeting neural inflammation to treat MND. So neural inflammation is more downstream of protein aggregates that Luke just touched. So as here, we discover we have a very beautiful illustration describing our current publication on cell. to take away the sting out of MND. And this is the way why we try to shut down neural inflammation. Because the role of inflammation in neurodegenerative disorders has been always a question, big question mark, because some people say it can be a marker of or consequence of neuron cell death, or it can be a trigger for that. So as an immunologist from WeHi Melbourne, we are very interested in the role of inflammation in human diseases. And then we start with MND as our very first neuron project. So I think, again, in this meeting, I don't need to touch too much about motor neuron disease. In general, it's a progressive neuron degeneration. Depends on where you come from. Some people call it MND, but some call it LS. But LS is the major form of MND. There's another answer for that. But what I want to highlight here is normally patients, they pass away within 2 years after disease onset. And it's due to there's no effective treatment or cure to reverse or to stop the disease progression. And also there's no reliable biomarker to identify people who have active disease beforehand. So they're making the survival window very short. So our mission here is to find to understand the mechanisms in this disease pathogenesis, to identify promising targets, and they provide a better opportunity for patient to survive and also for better treatment. So we know most patients in MND, they are sporadic. Only 10% they have a related family history. And that means everyone here, have a kind of lifetime risk or possibility to develop MND, unfortunately, due to at a certain stage of your life. But that's due to our living styles. So keep that in mind. Most of people, they don't have any family history to MND, but they develop MND. It's due to aging. And because aging are already being identified as a major risk factors, they impaired our metabolism and also to reduce our cell to function normally as we are young. So that will bring cellular stress and then they will cause damage. Toxins is another risk factors, and they come from smoking or infection agents. They will stress our system as well. And chemical insult from environment, and most importantly, genetics. So like what Luke already mentioned about SOT1, there are other genes has been associated with MND. So what I'm going to talk about today is the TARDBP, and they're encoding the protein called TDP-43. The reason for this, we are talking about this protein, is in nearly all LS patients or MND patients, they all have this protein aggregate. And this protein aggregate is pretty much composed by TDP-43. And then most of them are wild type. So that's the normal TDP-43. The reason for that is they have appeared in the wrong location and they cause cellular stress. Depends on the field of the research. Some people call it TDP for pathology, but for us as an immunologist, we call it proteinopathies. So it's a protein-related disease. So what is TDP-43? So remember the central dogma of biology, DNA and RNA are important to get the message to convert into something functional, which is protein. TDP-43 mainly sit in the nucleus where our DNA or RNA sits. So their major function is to function this balancing to maintain protein production and to maintain homeostasis for cell, for body functions. So it's very interesting. You can see TDP-43 is conserved across nearly all tissues. So not just in our brain as a CNS tissues, but also in the skin, gut, and also muscles. So this become very interesting question. People say, okay, you're talking about TDP-43 is the disease hallmark for the aggregate for MND, but why only affecting in the CNS, but not other tissues? So this might be due to neurons are more fragile in response to damage or cellular stress that caused by genetics or aging process. In terms of structure, in addition to DNA or RNA binding domains here, actually C-terminus is very interesting domain or structure because that's where most patient-associated mutation in TDP-43 being identified. And this mutation are still increasing by a lot of publications. But most important thing is this glycine-rich biochemical structure is a prion-like structure. So again, you know that from Luke's talk, this prion-like structure, they are prone to misfolded and they are prone to aggregate in the cytosol of the cells outside of nucleus. So what difference between wild-type TDP-43 and mutant TDP-43? It's a very interesting field because for a long time in medical research, we think wild-type equals normal function of protein, and that is related to healthy donors, healthy people, right? But mutation, it's related to disease. But in the case of protein aggregates, both wild type and mutant protein, they can aggregate and cause disease. So it's about the idea about the amount of protein stress that caused inside of cells rather than the type of protein related to the disease. So what they do in that is mutant are more aggressive in terms of misfolding and aggregation in the cells compared to wild type or normal protein. And once they are aggregate, it's very hard to degrade it. So our immune system cannot ignore such disturbance inside of cells. So there's a very good research years back from the state, and it showed the TDP-43 mislocalization into cells. And I see this from the patient samples, from the spinal cord samples. And this is the sporadic. So what you see TDP-43 in red color here, they all normal TDP-43, but in the wrong location. And most important thing for this paper is they found they're not just sitting there. Instead, they are touching a very important structure of cells in green color here called mitochondria. So mitochondria, we all know, is the powerhouse of cells that generate energy essential for cell functions. So when this protein aggregate called TDP-43 pathology, they invade into this mitochondria, they will for sure cause mitochondrial damage. So you can think about that. You lost your energy production, you will have a cell stress. So first, what is mitochondria? We know, in addition to powerhouse of cells, actually is the hidden bacteria living inside of your cells. And this is we call win-win or biosymbiosis process, because from the early life format during the evolution, bacteria, this type of bacteria, they invent into our cells. And then for some reason, they stay in there and become win-win relation. So just like a microbiota or commensal bacteria in our gut flora, they can generate some metabolites for our own good. So this is a very similar idea. But remember, it's a bacteria. So you can see they still conserve some bacterial-like structures. First, they have their own DNA. And this is very different from our DNA. So our DNA, you know, is from nucleus, and they form a highly arranged older structure called chromosomes. But this bacterial-like DNA in mitochondria is the ring structure, or we call circulate or supercoiled structure. This identifies they are still bacteria. And then because of that, they have a double wall membrane like bacteria to protect any contents in mitochondria or in bacteria to release or to expose to our immune system. So also mitochondria, they double like bacteria. So this is a very similar features that we have this bacteria, and under normal circumstances, they generating something we really needed. So it's for our own good, but it potentially is a bump inside our cells if something goes wrong like, for example, damage due to protein aggregate or stress. So this mitochondria has been identified as an inflammatory hub. They can trigger a lot of immune responses, and also when it's necessary, they will trigger cell death. So there's a bunch of studies in neurodegenerative diseases. They try to find or identify the role of mitochondria in the field, and we are one of them. So this is the illustration to show what will happen for mitochondria in response to protein aggregate, for example. Remember, they have that bacteria-like ring-structured DNA. Upon stress, it's well studied, they will release outside of mitochondria. And remember that normally we don't have a 3D floating DNA in the cells. So that will alarm or set off our immune responses. And we have a lot of sensors inside of cells to sense whether our cell is under attack by infection or whether we have a damaged cells, to generating some essential immune response or inflammation to begin with, as a protective mechanism. And normally we have this activation of immune responses is to remove or to resolve or to repair a damaged signal such as infection. But in neurons, this sort of inflammation is not good for neuron development. So this will become a chronic inflammation and then cause tissue damage. So our aim here is to study whether inflammation or this sort of inflammation signal is the cause of neuron loss in MND. So in the master lab at WeHi, we have a central hypothesis that within TDP-43 proteinopathy can trigger our immune responses that cause inflammatory cell signaling, and then it causes neuron cell death in MND. So the major aims, we try to identify what is the mechanisms underlying that neuron inflammation. So including what is the immune sensors for that and what is the cell signaling downstream for that. And this is a very good or interesting strategy because in signaling or cell signaling pathways always come with different potential therapeutic target. And we normally can decide inhibitors to target this pathway to inhibit these inflammatory pathways. And that's how we do for autoinflammatory disorders or how we treat cancers, for example. And then whether we can apply this knowledge from M1 to block neural inflammation to treat MND in preclinical mouse models. So just like what Luke showed in the cell models for the drug screen, we did a similar things. So we start with the cells, and then what we analyze here is using we call, for instance, microscopy to image cells. They express the protein of interest. So here we take the TDP-43 in red for what we make the wild type TDP-43 and also the MND associated mutations, and we express a lot of these proteins inside of cells and see what they change cell morphology and how they where they sit, because we can take that with the protein, for instance, protein, which is red color here instead. So in control, you can see mitochondria is very happy in blue. They form network. You can see this elongation mitochondria, and because under normal circumstances, DNA in green is sitting inside of mitochondria because they are protected. They just have their machinery to generate cell energies, right? But when we using a model that overexpress TDP wild type or mutations, you can see red, which is TDP-43 protein. They get inside of mitochondria in blue, and some of them release green dots outside of mitochondria, which is DNA. So this DNA is normal inside of mitochondria. Instead, it's got released. And mutant forms, you can see is more aggressive in the mitochondria. Also, you can see more 3D DNA got released due to mutation TDP-43. So this suggests the TDP-43 protein stress, they will trigger mitochondrial DNA release. And we confirm that by showing this as using this imaging technique. Then we're using these same models to generate more cell signaling data, but because of time, I can't go into that in very detail. But in short, we identified the primary immune sensors that sense TDP-43-induced mitochondrial DNA release, which is cGAS and STING. So in general, cGAS is the receptors that sense cytosol DNA or 3D floating DNA inside of cells. And then it's very interesting because you can see it seems very complicated, but they give us a very important message for treatment, and also biomarker. First, when cGAS, which is A receptors that sense or kept the 3D floating mitochondrial DNA outside of mitochondria, they will get activated and generate a specific messenger called cGAMP. Then this cGAMP will acting on another receptor B called STING. So in general, in the field, we just call cGAS STING pathways or cytosol DNA sensor pathways. Then upon this, and these all the two double checkpoints to make sure there's a freely unwanted DNA release due to protein aggregate or stress inside of cells by TDP-43. Then they will trigger cell signaling, generating inflammation in the cells. But so far, we are using man-made models that generating a lot of TDP-43 to force the cells to be sick. Can we see this in patient cells, which is more physiological? Unfortunately, it's really hard to get primary motor neurons from patients because I think it's a very painful and very non-feasible process to get it. But what we already have in the lab today is we're using sort of induced adult stem cell technology called iPSCs. So what we can do is we get the skin cells from the patients. Then we generate that, we will culture that in a dish in the lab, and then we give them a certain growth factors, hormones, or drug to push them and grow into mature motor neurons. As you can see in green color, they develop these beautiful neurons within a month. And then we are very excited to see that this cGAS STING inflammation in these three different patients with MND, but not healthy donors. Then we're using the inhibitor targeting this pathway. We have two different inhibitors. Remember, A is cGAS, B is STING. Then we can shut down this inflammation. So now the big question is, can we reverse neuron loss in MND by blocking these inflammatory pathways? So we treat these iPSC motor neurons with inhibitor against STING. Under microscope, you can see how a neuron degenerated from patients, and they don't really form those clusters anymore, and you can see neurons become more dimmer. But with 1 month in STING inhibitors or targeting these receptors in the pathway, you can see that although they're still very sick, but you can see these neurons or bundles, they are remaining. So that shows the blocking these neuron inflammatory pathways actually show less neuron degeneration. So that will be very interesting next to extend the treatment much longer and see what happens next. But I want to add is, remember this biomarker in this pathway, cGAMP, we are able to identify, or we are able to measure that and identify is increase in patients in neuron cells compared to healthy, but also we can find this in postmortem spinal cord samples from patients compared to another neurological diseases, multiple sclerosis, suggests this neuron inflammatory pathway is specific to MND. So to summarize this part of talk, we know TDP protein stress will trigger DNA and activating this DNA sensing pathway, trigger neural inflammation, which is very bad and play a role in part role to cause neuron degeneration. So based on this cell data, I think many of you will be interested in how about in vivo, like treating animals or treating patients in the future. So we have a preclinical mouse models they are very well and robust to recapitulate what happened in patients. So we're using a very famous mouse model that carry transgenic TTP43 human mutations, and they are expressed very highly in a lot of protein, mainly in the central nervous systems, in brain and spinal cord. So you can see these mice, these mouse, they will generate these neuron degeneration symptoms over the time, and then you can see this mouse start developing kind of we call duck feet because they cannot work properly. They kind of like crawling on the wall and the bottom of the cage about 100 days. And this video shows this about 150 days, they cannot upright themselves. And this is what we mark the endpoint of the disease development. But most important thing, whether they have that neural inflammation pathways we observe in cells, yes. So remember, cGAM is the biomarker for these pathways. And then when we take the spinal cord and cortex from this endpoint, there's an increased cGAM. So this is a very good model to start with, and then we can try to delete STING, take this honeybee STING away from MND. So we use genetic deletion to get rid of STING, which is in green color. This is the mouse model. They carry mutation in TTP43, but they totally lost the STING. And then compared to blue, which is just the disease mutation TTP43 model, you can see they protect the mouse to develop that they have a longer time they're taken to develop that neuron degeneration or loss of motor functions. So that's why they survive much longer, and then they behave much better, and they can still run at the same time point compared to these diseased mice. And just by grabbing their tails, you can see these TTP mutant mice are very sick. They cannot use their back legs. But when in those mice, they don't have a STING, they still have a very powerful back legs, and they're still struggling. So this is a very clear indication for that. But what I want to highlight here is the red lines. So we also have a model that only deletes half STING activities, and they also show protection. These data are kind of very important to tell us you don't need to inhibit these molecules fully. You just need to reduce its activity. You will see protection. And why it's important? Because when we need to treat patient later, we will use drug. But everyone takes drugs differently. Some people can absorb drugs 100%, some people do 80%, or even some people do 40%. But as long as we can inhibit this molecule called STING and shut down its activity, they will give protection for MND development. So this is again showing how their brain looks like in these mice. So you can see the sick mice, they lost a lot of neurons in the cortex in the brain. But when we have a half lost STING and fully lost STING, you can see more neurons. They are protected from loss. So finally, I would like to talk briefly about drug. So in cell, we have inhibitor targeting STING. Can we do this in mice? So when we let these mice to develop the duck feet, so that's the sign when they have a neuron degeneration. Then we treat them by injection of STING inhibitors for just 4 weeks to start with. So we are using the same protocol we're using in iPSC systems. And you can see the neuron is protected from loss compared to sick mice, and also their motor function or performance they test by behavior tests. So pretty much we put the mouse on the instrument, force them to run. So this can tell them how they coordinate their back legs and movement. So when they're treating with STING, they behave much better in terms of motor functions. So finally, we show, to summarize this talk, we show blocking these neural inflammation pathways as a very interesting and very promising therapeutic target for MND in the future. And this is recently highlighted by the New England Journal of Medicine, showing whether this can be the way to go as part of combination treatment in the future. Because we will just addressing the role of inflammation that play in part of neuron MND, but not full. As you can see in animal work, we cannot protect 100%. But before that, there are several hurdles to overcome. First, we need a biomarker to tell who those people in the MND population, patient population, will be benefit the most from inhibitor, for example, STING. Then that's why we are currently working on CGAP to validate that as a biomarker. And we're working on that to see whether we can identify or found that in the patient serums or cerebrospinal fluid, CSF, to provide a better painless or pain-free manner to quantify this biomarker for people who can use these inhibitors in the future. So finally, I would like to acknowledge everyone make this story and findings possible, especially my mentor, Seth Masters. He's been so supportive to allow me to do this neuron project for the first time in his lab. And then also all the collaborators and funding bodies, including MND Research Australia. Thank you for your time, and I'll take questions.
Okay, excellent. Thank you both, Alan and Luke, for that fantastic summary of your research. I mean, you've really done a great job there in translating some very complicated concepts and cutting-edge approaches into some simpler language that even people like me can understand. So, Luke especially liked the library and burnt hot dog analogy. That worked really well. So we've got a few questions lined up. So I'll start shooting some of those through. So one question now is, okay, this is a great question actually. Given the difficulties in diagnosing MND, when would these interventions, I think this is probably looking at, well, for both of you, when would these interventions be applied? And would they be successful after diagnosis, when obviously your disease can be quite far advanced?
Do you want to go first, Alan, or do you want me to go?
You can go first, Luke.
I'll go first. I'll go first.
Yeah.
Yeah, that's a really good question. And it's something that we in the field always keep in our minds, is that any treatment or intervention that we may want to provide people is likely going to be after after the disease has progressed far enough for symptoms to occur, which often means that you've lost most of your motor neurons already. I will say that the treatment that I described, the potential treatment, I should say, probably is likely to slow progression, but it probably wouldn't halt it. And it would probably, at the moment, we're thinking it's only effective for people who have the SOD1 associated MND. But given better biomarkers of disease, or even knowing, most people usually get screened for an SOD1 mutation. If they know they have it, these things could potentially be prophylactic. They could be taken in prediction that the disease might occur. But we still need to do many, many, many more tests before we know anything about how these will work in patients. Yep. Alan, thanks.
Yeah, Luke, I think wonderful. I think you give the best answer. Yeah, same as TDP-43. I think when these people come for before diagnosis, they get sick, right? So there is a lot of neuron cell death already happened. That's why in the field, they were talking in the early days, the most reliable biomarker in the field is called neuron filament. And it's due to, normally you can't measure that in the bloodstream, but when you see that, that means you already have a lot of neuron death in the brain. Same as what I'm talking about, CGAP. Of course, there's a possibility we might catch this CGAP production in the early days, but it's quite challenging because you don't know who is going to develop MND. So we currently use this as a biomarker for those people who can use STING inhibitor for potential treatment in the future. And also, again, blocking STING, taking STING out of MND is delaying the progression, not the cure. Yeah.
Okay, thank you for that. Just leading on from that comment, Alan, about CGAMP. Obviously, as you say, it's very difficult to do biomarker studies in humans from very early because you don't know which patients are going to develop the disease. But surely in your mouse models, you can get an idea that you could start testing levels of CGAMP very early and just see how it progresses as they develop the disease. Have you looked in your mouse models to see how early you get a signal for CGAMP?
Yes, I did. So we try to see the measurement of CGAMP over the time of mouse when aging, right? I can keep that in mind. These mouse models, they have tons of TDP-43 mutant TDP-43 in the brain, but we were not able to measure CGAMP by ELISA or by a very straightforward measurement approach before 90 days old. So we cannot. Maybe it's kind of like an under detection limit, but so now we are doing a more high tech, something called Simoa. So it's a small molecules platform to provide a 50-fold higher sensitivity than our standard platform. And then we have that now at WeHi, so we try to see that in mouse, but also most importantly, we are collaborating with Uni Melbourne to get the serum samples and CSF samples from sporadic and also familial MND patients. Yeah.
Okay, thanks, Alan. Here's a question for Alan as well. So if you've got, if neuroinflammation is a key player in MND, can sort of targeting more generic inflammation, such as even Nurofen or some of the more powerful anti-inflammatories, such as anti-TNFs, how come they don't seem to do much?
Well, so in general, we call it inflammatory responses, but there is a puzzle in terms of the cell signaling. So when the CGAS-STING pathway I'm talking here is not overlapping with the Nurofen, like a COX-2, COX-1, that like Panadol that targets, because those pathway they target is anti-pan or anti-hit, something like that. But for TNF, possibly, but immune response is very specific. So there are certain type of immune mediators we call cytokines. It's also different, depends on which pathway got turned on. So TNF is very general. It can be turned on downstream of by a lot of receptors, but CGAS and STING mainly turn on another cytokines we call type 1 interference, and then a little bit of TNF. And then in our current research, we know this disease is very much driven by type 1 interferon based, but not TNF. So it'll be interesting to see how the role of type 1 interferon downstream of these STING pathways and see how they protect LS or MND.
So basically at the moment then, you really need specific inflammation inhibitors you think are required for MND rather than the more broad spectrum ones that are currently out there on the market.
Yep, I think that's reflecting the current approval drugs, Riluzole, because that's the general antioxidant drug hitting mitochondria, and then they kind of slowing down, but because they don't target the particular pathways, that's why you still need to find the cause of the inflammation, but not in general. Yeah.
Another question for Alan, we'll get to you in a minute, Luke, don't worry. A question here about how do you inject STING or STING inhibitors into a mouse? Is it, do you have to go through a spinal tap, or can you do they can they travel across the blood-brain barrier quite easily?
This is a very good question, because when I present this work, people say, is your drug penetrate to BBB? Yes, so we're using this drug. They are published by several big lab and papers now. They can penetrate BBB, and also when, so you just pretty much inject mice IP or IV, you don't need to do a very high technical brain injection or spinal cord things. You can just inject it into your in their circulation. And just keep that in mind, if you have an inflammation in the brain or spinal cord, that kind of cytokines or immune response will make the BBB leakiness. So they will allow or provide a window for this drug or molecule get inside of the tissues. So which is quite safe, because normally our BBB is very intact, but only for certain people who needed this drug, they need it, and then they can pass into BBB.
Thank you, Alan. Actually, I think, Luke, I think we're funding a research project at Wollongong. One of your colleagues is looking at how to get drugs across the blood-brain barrier, but we'll leave that for another talk. Luke, I have a question here about, are there any sort of studies in progress to try and find out any biomarkers or early ways of detecting aggregation and protein misfolding in the brain?
Yes. So researchers around the world have realized something about the disease and many neurodegenerative diseases, and this is that the aggregates that form in or outside cells are capable of replicating themselves. They can take healthy protein and add them onto themselves and grow in size. And so one of the things that was found is that this can be exploited by us to remove CSF from someone and to then add the tiniest, smallest amount of CSF to a little test tube with a form of say, for example, TDP. We can make TDP in a laboratory using bacteria. And if we mix the tiniest little bit of a patient's CSF with that TDP protein, we can monitor whether or not the TDP from the bacteria will aggregate with the CSF from a patient. And this is surprisingly sensitive. It's in fact, recently this technique sort of taking off. It was first found in the prion field, and it's potentially going to be something that can help us make early diagnosis. But again, it's one of those things where we need access to CSF, which is painful, but I'm willing to bet most people would want to do that to figure out whether or not they have MND or not. But we are still looking for serum blood-based biomarkers too. Yes.
Great, thank you. And we have one question here. It's slightly unrelated, but see how you go. How does riluzole work? And does that relate to any of these two pathways that either of you two have talked about?
Alan, do you want to do this one or me? Do you want me to do it?
I think it's more short one, right? So I'll leave it to you.
Yeah. So riluzole is one of the only FDA-approved compounds for MND. And the way it works, or the way we think it works, because we don't know 100% yet, is that it blocks the neurons from letting out a molecule called glutamate. And so glutamate is a very, very, very small molecule, and it's part of your cell signaling in your central nervous system. And one of the features of motor neuron disease is that before your motor neurons die, they become hyperactive and they release a lot of glutamate. And this in of itself is toxic. So it's what we'd call very downstream from the initial cause of MND. But it's one of those things where we can block it and sort of give someone a little bit more time, slow the progression down a little bit. And so that's how riluzole works. It prevents this, what we call excitotoxicity from occurring.
Yeah, can I just quickly add something? I think I make a very kind of incorrect statement about the drug. What's the drug? Riluzole? Yeah, because I thought it's targeting the antioxidant, but that's the radical, right? So another newly approval drug. Yeah, sorry about that. Yeah.
Okay, no problem. Okay.
Yep.
Thank you both. I think we've reached the end of our time slot now, and I'd just like to thank you both again. I thought that was really fantastic. Like I said, these are really complicated concepts and real cutting-edge stuff that you're doing there. And it's great to be able to take this to the general community and hopefully give all our all the MND patients and carers and family members out there a better idea of where we're going, and hopefully some hope as well for the future, that there is some really great research being done out here. So we're going to keep this program going this year. It'll be pretty much roughly monthly. Our next presentation is during MND week on the Wednesday, the 5th of May, when we'll have a couple of our researchers talking about genetics and how that relates to MND. It won't be the exact same day every month, so please keep an eye on your emails and our social media, etc., and you'll get plenty of notice for when our next state of play is. Everyone who registered will also get an email with a link to the recording. If you didn't get a chance to watch it tonight, then obviously you can watch it at your leisure, and feel free to share it through your networks. I think this sort of fantastic talks need to get out there. So that's it for tonight. So I'd just like to say good night. I'd also like to thank everyone else who came on to watch these talks tonight. I hope you found them as good as I did. So good night to everyone. And